Okay, so I'm going to introduce online speaker, Dr. Tal, Unraveling the Immunopathology of Infection-Associated Chronic Illness, Sex Differences, and Predictive Diagnostics. And the biography of Dr. Michal Caspi Tal, she describes herself as an immune engineer. She is principal scientist at MIT in Boston, and she leads the Tal research group within the Department of Biological Engineer, also serves as Associate Scientific Director of the Center of Gynopathology Research at MIT. So she works in a number of different conditions, from tick-borne diseases to COVID, and similarly, as I've mentioned before, there's lots of similarities between these infection-associated chronic illness. There's also important sex differences in the immune response to infection, which could impact on the risk of developing chronic illness. So Dr. Tal is running a large clinical study at MIT called Maestro on chronic Lyme and long COVID, and complementing that with the use of the mouse model of Lyme disease to deeply investigate sex and age differences in the immune response to infection. Her research is focused on developing predictive diagnostics to distinguish those at risk of developing infection-associated chronic illness. So welcome, Dr. Tal. Thank you for coming.
Thank you so much. And I'm so sorry that I'm not there with you guys in person. It's been an amazing meeting so far, and I've been enjoying it from my office. So I'm going to take us right in. We have three main learning objectives today. I'm going to tell you guys about our approach to predictive diagnostics. I'm going to walk you through some of the basics of our mouse model, and also some of how we're approaching our clinical study, Maestro. I have no conflicts of interest to declare. And there have been such great talks on Lyme already. The only thing I want to try to convey from this slide is that there is a life cycle for Borrelia, where it is a beneficial infection in the ticks, and it is using the mammals as a reservoir to infect a new crop of ticks in the spring. And in these mammals, Borrelia is really causing very minimal disease in general in its normal animal reservoirs. We are an accidental infection. And so that's really important for how we think about both our mouse model and what we're learning from that, as well as how we're thinking about the immune response to infection. Okay, so I always start off with this slide, and I feel like with this audience, we've had so much discussion of it, but since there was an earlier talk about the use of the term chronic Lyme, I did just want to, touch on how, for a long time, this was treated as very different from other infectious diseases. But, as you're going to see, I really don't think it's that different. And I think that it becomes really critical for us to understand we have to understand if the disease is coming from the actual bacteria or from the immune response to the bacteria. So we'll talk about that. So I asked Dolly to make me a Rubik's Cube that says infection-associated chronic illness, and this is what it had made me. And I thought it was perfect because I thought it actually perfectly conveyed where we are right now in understanding this, where there are some parts that we think we know that are wrong, and there are some parts that we can almost just see how they're going to come together. And the three main questions that guide my lab are: why doesn't everybody bounce back from an infectious disease? When most people recover, why are some people not able to recover? Why do the infection-associated chronic illnesses skew female? And how do we distinguish between protective and catastrophic immune responses to infection? And you're going to see that we are really using both the mouse model and our clinical study to try to address these questions. And this is the updated new model from Gemini Nano Banana. I asked it to remake me that cube, but to put in some of the pieces that we're starting to see, like blood flow and loss of tolerance to cell flora, or allergic responses, or barrier dysfunction. So we're making progress. Okay, so as an immunologist and as an immunoengineer, this is really my view of the immune infection interface. And so we have your immune system. It is trying to kill invading pathogens and trying to prevent them from replicating. But in doing so, it is directly causing disease. The pathogen is also directly causing disease. And so our biggest question really becomes, in this particular scenario, who is causing the disease? Is it the immune system or is it the pathogen? Because as we've been talking about, this is the difference between immunomodulators or antimicrobials. And so it really makes a huge difference for treatment. And the case I want to make to you guys is that as we've been doing diagnostic serology, even the more advanced diagnostics that you guys have been hearing about today, we are still essentially counting IgGs or other bulk isotypes of antibodies to ask the immune system a yes or no question. Have you ever seen this particular bug? Yes or no? And the pitch I want to make to you guys about predictive diagnostics is that we can ask the immune system a more open-ended question. If you saw this microbe, how did you respond? And use that to really map out illness trajectories. And so how would that work? So the concept is that we really have to flip around how we think about the antibody. Antibodies have both the part that can bind to the microbe, as well as this part that you're now seeing flipped to the top, which is the part that can actually bind to immune cells that have these Fc receptors and have different immune effector functions that we actually know quite a lot about. And so the idea behind this concept is, let's use what we know about these FCs, about these portions of the antibody and which immune cells they trigger responses from, and let's try to build these illness trajectory maps that really allow us to profile what aspects of the immune system are getting involved in the response and how is the immune system responding. And so this is this concept behind what we call the flip, which is this, both around this, flipping how we look at the antibody, but, using flow cytometry-based immune profiling so that we're not limited to looking at just one type of antibody. We can use the Borrelia itself as a bait to profile all of the antibodies that are binding to that Borrelia and look at them as ratios. And so, I used to really look to strep as an example of how we could move forward in this field with very aggressive diagnostics and treatment strategies that have made rheumatic fever an extremely rare disease now in the US, while it used to be very common and remains common elsewhere. However, if we think about PANS, if we think about other outcomes that we can get from strep, and how many people don't actually need antibiotics to strep, whether they're the 20% of people who are strep carriers, or whether they're the 70% of people who will just get strep throat, have a response, a few days of a sore throat, and they're already recovering by the time we give them antibiotics. And what became really clear to me is what we need is a better way to ask the immune system how it's responding and to really be able to intervene in a very serious way when the immune system is responding in a catastrophic way. So this is actually an image from a review that we put out in the early days of COVID as we were doing our salivary antibody immune profiling, and we were trying to make this case around the vaccines and how they were targeting systemic immune responses against circulating components, which are very important for protecting your vital organs, but they weren't protecting us at the barriers where we encounter these infections. And so this is just to get across this concept that across different infections, it's really important to think about what type of immune response you need to elicit, where you need to have protection, and really to be thinking about antibodies not as a one size fits all, or, looking just at IgG will tell you something meaningful about other isotypes that might be more important in the response, like in this case, IgA. And this is to show you, Alison Redman and John Ockett have been doing some really beautiful work at Johns Hopkins, looking at Lyme disease and immune responses to Lyme disease over many years. And what you can see here is that there are important sex differences in the diagnostics that we're using. And so as you know, as poorly as the standard ELISA performs for everybody, it's performing even worse for women. And that is something I think we really need to be thinking about and trying to understand why that is. And so I love this cartoon because it really displays the concept that I think about in this way. And this is really this, the tool of predictive diagnostics is to use the fact that we are already tagging the microbes that we see, and we can just we essentially can just look and see how the immune system is already tagging them to really get a good understanding of how the immune system is responding. And so this is just to show you that we make a whole bunch of different kinds of antibodies. And the way that we have been looking, perhaps at IgM if we're looking very early in, or just at bulk IgG, we really miss this very critical resolution between different IgG subtypes. And that tells us a great deal about how the immune system is responding, as well as other isotypes, like in this case, IgE, which will be important for some of the things that I'm going to talk about, such as mast cell degranulation. So again, this is how this assay works, where we actually take the same Borrelia we use to infect our animals. We use that as a bait to pull out all of the antibodies that bind to Borrelia and look at them by different ratios. And this is just to show you some images of what it looks like if we then look at them under the microscope. There's actually a lot of beauty in there as well. These are Borrelia under attack by different antibodies. So the concept essentially goes like this, that we can map different illness trajectories if we use predictive diagnostics. And I'm showing you a whole lot of different kinds of catastrophic immune responses that we are looking for. And what I want to show you here, so this is from our profiling of samples from Lyme Biobank. And what you're seeing here is somebody who is over a year past their doxycycline for Lyme disease and never recovered. And what you're seeing is they're actually making a ton of IgG1, IgG3. They're making a bunch of different antibodies. And I do think this could help us rethink about IVIG, which is mostly IgG1, and then IgG2, and then less IgG3 and IgG4. And I want you to see what somebody looks like who is well on their way to recovery. And what you're seeing here is that this person is making a ton of IgG2 and IgG4. And this is going to be really important in how I'm thinking about this. I want to show you, this is this schematic that we've been working off of, that these, heavy on the IgG2 and IgG4 surprisingly seems to be protective and important in recovering from Lyme, as opposed to having a response that's very heavy on these more inflammatory types of antibodies. And so I just wanted to pull out a very, very recent paper on bioRxiv. You can see from just a couple weeks ago, looking at long COVID. And I was so incredibly pleased to see that they're seeing really similar IgG subtype ratios that we are, where this high IgG4, IgG2 ratios are actually protective and more seen in the people who go on to recover, as opposed to the people who are going on to develop long COVID. And what I think is really important about that is that this is going to really better clue us into what's happening, and also allow us to track this better in people who are suffering. So this is just looking at more of these illness trajectory maps here. And the thing that I want to pull out for you now is to show you that a third of these people were making pathological amounts of an antibody type called IgE, which is this allergic type antibody. And that begs this question, can you become allergic to certain bacteria. And in one of my questions for one of the amazing talks earlier today, I was bringing this up in the context of what about the spirochetes in your mouth? What about other spirochetes that you encounter? What happens if you become allergic to a bacteria? This is something we're actually very well poised to investigate in our mouse model. So in our mouse model, we compare C3H mice to black 6 mice. There are some very important differences between these animal models. I do want to mention that C3H is both the standard Lyme disease model because they develop a pronounced arthritis and carditis. They are also the standard peanut allergy model, and it is actually because they have some pretty bad intestinal permeability. And I can tell you that that gets even worse with the infection. No model is perfect. There's some things that aren't great about the C3H, and there's also some things that aren't great about the black 6. But in general, the black 6 are considered to be more tolerant to Lyme disease, but that's only if you ignore the gynecological diseases that we have recently reported. So I wanted to show you this animal. This is actually the animal. This animal changed my life, to be honest. This is mouse 23 from our male versus female experiment. This is an almost unrecognizable uterus here with just unbelievable amounts of Borrelia inside. It's completely swollen beyond proportions. And this is showing you for reference on the right, a normal uterus to compare that to of an uninfected animal. And this is actually how I ended up at MIT Center for Gynepathology Research because I just really had to understand what was happening here. And we have a really lovely preprint under this, which is going out very soon for review by Paige Hansen, Colburn in my lab about how Lyme disease increases risk for multiple gynecological conditions. So definitely check that out if you're interested. And I have a postdoc in my lab, Guido Passani, who is following this out and is now doing MRIs on our animals so that we can follow this in vivo and check out different treatments. And it's becoming a very, very useful model in the lab. And just because there were other talks mentioning these lymphadenopathies, I did want to mention that Guido is following out how the draining lymph nodes of the uterus are showing a very long-term ongoing reactivity, which we're following as well. So stay tuned for that. I want to circle back to the C3H mouse model, and I want to say what an inspiration Anne Maitland has been on this, where she, was telling me it all starts at the barriers, and I have really come to think that that's a huge piece of this. And I wanted to bring up this point that, our immune system, well, it's made to shift away from how it responds to an acute infection to a more exhausted response to a chronic infection. When you have a wound, when you have an open wound, or when you have these open barriers, the immune system is not able to shift gears away from this sticky, clotty, even fibrotic response that is meant to close wounds, and I suspect also compromised barriers. And I do think that's really part of what we're seeing. I told you I would go into this IgE piece a little bit, and what I wanted to show you here is that mast cells have high affinity IgE receptors, and our C3H mice are this very, allergically prone animal model. We can look at this in them, and we have been developing this mouse model for a while and following out these different features of it. We have a really nice paper with Hannah Lila looking at a secretoglobin that's actually very important in the response to Borrelia burgdorferi. But what I'm going to talk to you about now is this study that we had where we were following out the development of atopic dermatitis in our C3H mouse model. So this work was done by Brandon Lee in my lab and Xingming Feng. And what they noticed is that if we left the mice infected for 2 years, they developed a very profound atopic dermatitis of the tail, and that if we infected 2-year-old mice or even 1-year-old mice with Borrelia just for 8 weeks, we could recapitulate this very profound response. And so you didn't need to be infected for that whole time. You needed the age plus the infection for this to happen. And as we looked closer in, this is looking at cross sections of the tail. What you can see is that this really is an atopic dermatitis. There's this hyperplasia, there's this keratosis and spongiosis that you're able to see, especially in this bottom right infected 2-year-old tails. And so we use Borrelia that have been genetically engineered with luciferase so that we can see them light up and tell us where they are. And what you can see is you can watch the Borrelia spreading throughout the body, and you can see that they are going to the tails. So we know that the tails are infected. And then we see that the C3H have more infected tails. And then we know that they also develop this more profound atopic dermatitis. But what we saw, and this is a story that's still unpublished, but is currently in progress and will hopefully be submitted soon, is that if we let that atopic dermatitis keep going, they will develop a very profound hypermobility such that we can tie the tail in a knot like an EDS model. But to remind you, these are wild type C3H mice with no known collagen gene defects. So this is a non-genetic acquired hypermobility following the Borrelia infection that does not appear in uninfected animals. We did show in our atopic dermatitis paper that atopic dermatitis is being mediated by mast cells, and mast cells are coming in and degranulating. And what we can show in our hypermobile C3H mice versus our non-hypermobile as long infected black 6 mice is that we're having a very different immune response. So in the C3H hypermobile mice, we have all these mast cells infiltrating in. In the black 6, it's macrophages. These gorgeous images are also label-free and allowing us to look at Borrelia in the tissue, and that's all work coming soon by Kunzan Liu in Sishen Yu's lab at MIT. So our MIT Maestro study, in my last few minutes, I'm going to take you through this and what we are really trying to use this to understand. So we are comparing acute infection with Lyme disease to people who are over a year not recovered from Lyme or over a year not recovered from COVID. And they are all getting compared to recovered controls. And we are doing everything that we can possibly think of. So they are completing extremely comprehensive surveys. We're doing physical assessments when they come in person. We're doing a whole suite of neurological and neurocognitive and blood flow testing, as well as hypermobility testing, spine assessments, skin barrier testing, and a whole slew of paired biological sampling, including blood, throat swabs, saliva samples, sweat, urine, vaginal swabs, rectal swabs. And the concept that I just wanted to convey here is that when we lean on patient-reported outcomes alone, patients can only, our participants can tell us how they're feeling. And when we use these in clinical trials, I just want to point out the concept I was trying to convey here is if there's structural damage, how do you ask somebody if their connective tissue is damaged and if it's healing? And so what a person can feel and what's actually happening underneath, these are so important that we find ways to measure. And so I really think it's important that we design our studies and our clinical trials in ways that we're really measuring everything that we possibly can, and that we're thinking really hard about our exclusion criteria, because, well, you can make things simpler by, excluding comorbidities. The comorbidities they're really just part of this. And I think we have to embrace that. So taking a look back into these kind of catastrophic immune responses that we want to profile, I'll just quickly take you through and kind of try to highlight for you that what we are trying to do is ask our participants all of these different things. So we have our participant reported outcomes. So we have what they're telling us. And then we have all of these different measurements that we are conducting either during that clinical visit or on the sample profiling that we're doing on those samples afterwards, where we can then compare these different measures. And so we're very interested in the gynepathologies and this hormone immunity axis, the neuroinflammation. And so some of these are just teasers here of some of the data that we're already getting. We hope to have the study completed by the end of this year, but there's already data that is just so profound, even before we've finished our cohort. So this is showing you this eye tracking data, and you can very, very clearly see differences between people who have recovered and people who have not, in addition to their own patient-reported outcomes, as well as measures that we have like WAVI. I'll quickly show you. WAVI also gives us these maps. So these are these EEG caps that you wear. You can see, lights on when they first came in, and 6 months later, as they're developing these symptoms, you can see huge differences in these voltages. We can look at mitochondrial, metabolic dysfunction, energy levels. And this is where we're starting to see some really interesting differences between long COVID and Lyme. And we're able to see some of the things that are being reported for long COVID right now, like this difference in N-acetylmethionine. We can look at barrier dysfunction, motility, microbiome dysbiosis. So we can use a Nevisense, look at impedance metrics where we start to see some differences. One of the most profound things I wanted to tell you guys about is that when we look at blood flow, vascular dynamics, coagulopathies, we have a lot of different ways that we're approaching this, but one of the most profound is the capillaroscopy. This is showing us things that we really think will need to get integrated into clinical care that could be very important. And I just wanted to show you how dynamic this is. So this is where we're doing capillaroscopy before and after the Nasoline test. And on people who are symptomatic on the Nasoline test, we can also see huge differences in their capillaroscopy. This is a non-invasive, not painful. This could be used in children. I really think this is just so critically important, and I wanted to make sure to say that, and I know I'm running out of time. I'll just say one last thing about how we're looking at pathogen persistence. So we have actually, originally we were planning to do Carius on our entire cohort, despite the price of that. We are switching to doing 30x whole genome sequencing on everybody and doing that ourselves, because the problem that we had coming out of Carius, and here I'm just showing you a sample of our Carius data, is we had so many people with no call. And the problem is that, places like Carius, they only report to you things that they find that they have not found in healthy individuals. But because we want to understand burden of different, EBV, CMV, things that we are expecting to see also in healthy individuals, but we want to see if they're different, we actually are thinking that we are going to have a much easier time of doing this ourselves with deep whole genome sequencing instead, as well as RNA sequencing. Yeah, I'm going to have to skip towards the end. I just want to say, because we have such an incredible crew here, even though I'm joining you from online, I'm going to show you my email. Please reach out. I would love to connect with you. And if you have a hypothesis that you are burning to test in our Maestro samples, then please let's talk, because we really want to collaborate. Our impact statements and references, and I can share these slides. At MIT, we are launching a menstruation science initiative that we are very excited about. And I just want to thank all the people in my lab and at MIT who have helped us so much with this. We work very closely with Linda Griffith and Doug Laufenberger, as well as the Center for Clinical and Translational Research at MIT. And I didn't get to show you all the incredible label-free imaging from Sishan Yu's lab, but I did get to tease it a bit, so you can see that that's going to be amazing. And here you see my email. So please do feel free to reach out, and I would love to take any questions that you have.
Thank you for the amazing presentation. We do have questions.
Hi, thank you so much for this incredible research and presentation. Just truly incredible. My question is, we see pockets of EDS diagnosed without genetic backup in human studies, and it kind of always comes from a certain geographic locations, which is coincide with the hyperendemic areas of Lyme disease and co-infections. So has anyone in your animal model group ever correlated this to the human studies?
That this is a huge goal of ours that we are trying to take on. We are so excited to be able to recapitulate this in the mouse model where we can really start to take on some, mechanistic testing of interventions, right? So we can test, as I was showing that we could test Xolair, we can test different mast cell stabilizers, we can test duplicescent. So we have a way to try to, from the time we first see the atopic dermatitis and to try to intervene before it develops into that full-blown hypermobility. What I didn't have time to show there, but we are about to be publishing is that hypermobility of the tail is also correlating with hypermobility of the spine. And so these tails and spines are getting stretchy because the connective tissue holding the vertebra together is getting compromised. And so we are very, very interested in following this in humans. So in our clinical study, we're doing the Baton score, we're doing other measures of hypermobility. We're also looking at, subluxations and other things like that, that we're trying to look at more at an epidemiological level. But yeah, I'd love to hear more of your thoughts. If you want to, write me, I'd love to talk more about it.
I have another question.
Yeah.
Immune senescence. Do you have any measurement of the animal model immune senescence for their autoimmune or reduced immunologic response on the mouse model to prove immune senescence? Compared to COVID versus Borrelia and co-infections.
Yes. So what we have is we have our comparison between the Black 6 and the C3H. And the Black 6 are making a very different response. So they're making this very macrophage-heavy, neutrophil-heavy response, where the C3H are making a very mast cell-heavy response. The problem with the Black 6 response is all that neutrophil nets, they're actually clogging up the lymph nodes, and they're actually creating a situation where we consistently measure, and others have as well, this actual hypoglobulinemia, where the follicles aren't developing properly, and we're not getting this great, isotype switching in the Black 6. We're not getting great antibody responses. We actually get much more profound antibody responses in the C3H. Pathological antibody responses, but much, much more than what we see in the Black 6. The Black 6 are walking this very fine line between the Borrelia are behaving differently, they're being very immune evasive, and the immune system is really ignoring and dysfunctioning in how it's responding, but in a way that's actually at a total level causing much less damage. It is causing quite a bit of gynepathology, but they're not falling apart the way the C3H mice are. So we are very much able to follow that in our animal model. We are able to see development of autoantibodies. We can see that also in our human study, and we're measuring these different cytokine ratios that we think are extremely telling for how the immune system is poised, and these FC ratios as well. So I think we are going to learn a lot about how the body is trying to walk that line between ignoring a pathogen it can't clear and, causing too much damage, and both come at a cost.
Thank you. There's just one online question that we have to take. The question is, have you looked at the HLA for the recovered versus unrecovered?
Yeah, so there have been HLA findings that we have made together with Hannah Lila when she ran the GWAS for Lyme of HLA DRB1. Others have previously reported, there's been a couple, that HLA has been reported before as well as others, and we are very much following that to try to understand what is the epitope that is getting displayed there and why that HLA is especially risky, and could that be used actually, could we just kill off the T cells that are seeing that HLA in that context if that's what's causing the problem there. So that could be a therapeutic approach, and that's very much something that we're paying attention to, especially on HLA DRB1. Yep.
Great, thank you.